Yeah.
Yeah, I was about to say I'll introduce you, Jacqui, but I probably don't need to because I think everybody on will know exactly who you are. But
Do a
just to
quickie.
say a massive thank you before we start again for doing this whole series of CPD for us. Um, uh, I feel like you must spend an awful lot of time delivering webinars.
Stuck in my freezing dining room.
Yeah.
I've got
There
my
are a few more
blankets now.
people.
Like,
There are a few more people trickling in now, but thank
yay!
you very much. We're really grateful to you for giving up your time.
No problem.
And so yes, I will let you. I will let you carry on.
Cool. Thanks for coming, everyone, tonight. So we decided, I can't remember what we did before with the vets, because it was 2 years since we did our last set of webinars with Excel vets, but we thought we'd have a chat about detecting and dealing with anthelmintic resistance in practice. So for those of you who have maybe forgotten their parasitology or weren't very interested in the first place, I'm just going to go through a little bit about the parasites that we have to think of as threats in horses, and then talk a little bit about resistance and why it occurs, and then talk a bit more about, well, how can we either detect it and deal with it in the field. So, as I'm sure you all know, the most common parasites of horses are the cyathostomins, or also known as small strongyles or small red worms. These are pretty much ubiquitous parasites across the world. Most grazing horses have infections with these worms at some point in their life, and horses tend to develop a slow immunity to cyathostomins, so that what we find is that younger animals tend to have these larger burdens, and as animals get older, they tend to be able to regulate the parasites better and have lower burdens. That's a general kind of trend. I mean, that is a bit of mucosa we've got stretched across a microscope, and it's just been photographed actually, and you can see each of these little spots on it like a blackhead is an encysted stage developing L4 cyathostomin, and that horse, when we enumerated it, it died. It had larval cyatostominosis at something like 3 million, over 3 million worms that we estimated within its mucosa. So you can see this is a parasite that we really need to make sure that we keep on top of, primarily by good management, so that these burdens don't accumulate. The next parasite, for those of you that have had some pasta to like me tonight, I always think these tapeworms are quite like conchiglie if you stretch them out. And this is a common parasite, although its prevalence does vary from site to site. In some groups of horses, we see a prevalence of over 80% of Anapocephala perfoliata, but in other groups of horses, it's below 10. In most cases, it's got a relatively low prevalence in groups, particularly if the pasture management is good. The other parasites that we need to be concerned about in horses are parascaris infections and foals, as you know. That's like these big spaghetti or noodle-like worms here, which live in the small intestine. And these are really common on stud farms. The parasites are nematodes like the small red worms, and the eggs that the females reduce in the millions are very robust, and they can hang around in paddocks for a long time. The other parasite that we should probably be concerned about more from a sort of crystal ball perspective are the large strongyles, particularly Strongylis vulgaris. And at the moment, where microcyclic lactones have been used, these parasites are pretty rare. But the work that's been done, twofold things they found. First of all, where you reduce treatments, particularly in areas such as Denmark, where they're strongly following diagnostic-led worming programs for many years now, they've seen the prevalence of Strongylis vulgaris increase. There's no indication yet that that's been associated with an increase in the incidence of clinical disease due to the parasite. The other thing is work that we've done and others have done is actually if you're looking in enough depth at the parasites that are present within populations of horses, you do find large strongyles and you do find S. vulgaris. So it's like it bubbles along there at low levels, and we just have to be careful where we're not using anthelmintics and we've got open herds, for example, and we're not checking that it's there, that it can actually increase in prevalence and abundance to cause clinical disease. Now, with all of these worms that I've spoken about, it's really important to keep in mind, which I'm sure you do know anyway, because you're dealing with horses day in, day out, and they're not all lying dead with parasite infections, is that most infections are subclinical with helminths, and disease only occurs really when the burden of that parasite is high within the individual. And these different worms can cause mostly gastrointestinal associated disease and clinical signs such as weight loss, colic, and diarrhea. And keeping in mind that Parascaris itself is a respiratory migratory worm, and it can cause respiratory signs in young foals with heavy infections. Now, someone did ask about treating horses and choosing wormers for the um, individual horses. So this is a bit of an introduction to that, and I'll come back to this later on. So despite the fact that we've had all these classes of anthelmintic for over 30 years now, we really haven't had any new compounds coming to market, and there doesn't seem to be any new actives with new modes of action coming down the pipeline, partly because it's very expensive to develop these anthelmintic products, and the horses are really treated as sort of minor species in the general sort of pharma community. So I'm not holding out for any new anthelmintic compounds coming to be used in horses in the foreseeable future. So it means what we've got is we need to preserve the anthelmintics that we've got, which are three classes for the treatment of nematodes, as you know, the BZs, tetrahydropyrimidines, and the macrocyclic lactones, ivermectin and moxidectin. And for cestodes, tetrahydropyrimidines used at a double dose, in the UK, that being Pyrantel, Embonate, and the isoquinoline, pyrazine, praziquantel. Now, in terms of the efficacy, most of the nematode compounds work against the parasites I mentioned if the populations are susceptible, but we do have a few complexities in that the cyathostome and encysted larval stages, which are shown in this slide, which comprise the early L3, the late L3, and the developing L4. These appear to be less susceptible to the effects of anthelmintics, and the only two preparations that have licensed efficacy and against these three stages in the UK are a 5-day course of fenbendazole at the standard dose rate and moxidectin as a single administration at the standard dose rate. Pyrantel, a single dose of fenbendazole, and ivermectin seem to have less effect against these encysted stages. When they were originally developed, they had good effect against all the luminal stages, including larvae and adults, and in some cases, the later stages of the mucosal larvae. If we were to, for example, perform a fecal egg count reduction test in a sensitive population of worms, and we're looking at strongyles in this case, but also ascarids as well, you would expect both fenbendazole and pyrantel to reduce the egg count span, a mean egg count reduction of over 90%, well in excess of that. And if you were to do the same sort of experiment with ivermectin and moxidectin, you would expect both those compounds in a totally sensitive population to reduce the egg counts in excess of 95%. And when the compounds first came out, it was pretty well 98, 99% consistently identified. And on the basis of the efficacy of these actives, even from way back in the 1960s, and it was the same advice that was given for both cattle and sheep as well as equids, was actually just to treat all animals at regular intervals with these wormers. And they defined the interval to do the treatment frequency based on a standard egg reappearance period of the compounds in drug susceptible populations. So say the egg reappearance period, for example, first identified for benzimidazole was 6 weeks. They said, well, actually just come in, treat with benzimidazole every 6 weeks, and what you'll do is you will suppress egg output, no eggs will go in dung, and therefore you won't contaminate your paddocks, and your paddocks will become parasite free, and your horses won't get any infections because the paddocks are parasite free, and your worms will be eradicated. Well, sadly, that is not what's happened, and we've failed to eradicate parasites using these interval treatment methods, and all we've managed to do is very nicely is to select for anthelmintic resistance in worm populations of different species across the world. And this is just a little cartoon that sort of gives you an indication of how this selection works. And what we know now is that obviously helminth populations, especially in susceptible animals on contaminated grazing, can be enormous. They can be to the order of millions of worms. If you think when we've got enumerated parasites, small red worms from heavily infected horses at the abattoir, and in individual horses we've counted over 6 million worms, you can imagine that's only a small proportion compared to what's on the paddock. So these populations can be absolutely enormous. And what we subsequently found out is that helminth gene sequences tend to be variable. So the sequences of the target genes of these particular anthelmintics, the target sequences of the molecules to which the compounds attached or had their effect, they were also subject to variation, so that we would have the situation where a large population of organisms with genes that were quite variable, you apply a strong selection pressure, especially if you start to look at something that's about 98% effective, which was the efficacy threshold for registration for these compounds. You apply that selection pressure, and what you end up is with a subpopulation of surviving parasites that tend to have the gene alleles which contain the polymorphisms that change the shape or the configuration of the molecule, and then it renders that parasite less sensitive to that compound. And that's what we found in cattle and sheep and horses and humans and dogs and cats, I'm sure as well, that as you've applied these compounds very widely over time and with repeated treatments, we continually select for these parasites which are less sensitive to the drug, and they become the major part of the population, so that your male and female worms mate, or in the case of tapeworms or flukes, for example, the resistant worms survive, and the progeny are then more resistant than the previous population. And over time, this has led to the problem of anthelmintic resistance in just about every parasitic helminth that we are aware of, with a few exceptions. Now, in addition to the sort of background to resistance and repeated treatments, what are the other things that select for resistance? And it's things that some of us, if we're older, would have been taught at vet school to do, and things that probably go all the time out the field. So obviously the regular treatments in a whole group of animals would be a very strong selection pressure, because at that treatment you're not really allowing any parasites within the overall population to escape the effect of the drug, and you're concentrating therefore the subpopulation of less sensitive worms that survive that treatment. Something that was really a great idea, actually, if the wormer worked 100%, is dose your animals, give them a day or two, and then move them on to clean grazing. But what is quite apparent is if you dose your animals and you've only got, in a couple of days, there's the surviving worms from the treatment left, it means the surviving worms will mate, and the parasites that are going on to the paddock will be from those less susceptible worms, and then you'll have a higher concentration of resistant alleles in the clean grazing than you would have had on the previous dirty grazing. So we don't do that anymore. Underdosing is an obvious thing that selects for resistance, because what you're doing is you're exposing the parasites to suboptimal levels of that drug in situ in the host. And that means that you're more likely to select animals that might be slightly resistant compared to ones that have a sort of more higher level of resistance. So you're promoting more resistance in terms of exposing those parasites to less drug and doing a bigger selection across a range of worms with different levels of susceptibility. Again, treating the wrong target species with a compound. Hopefully this isn't done as much as it is in the past, but I have come across people that do it, maybe give something like doramectin, or to save money, give a cattle ivermectin to a whole group of horses, particularly an ivermectin that's for injection, given orally. These things will all lead to the suboptimal pharmacokinetics within that target host, and it will lead to a suboptimal level of wormer around the environment of the worm, and therefore be another driver to select for resistance. Now, the problem with resistance, in addition to this background, is that it's not phenotypically obvious. Horses don't walk about with a sign saying, I've got drug-resistant worms, and even fecal egg count reduction testing doesn't detect resistance that early in its development within a population of animals. Now, there's not been really anything done on horses in this, but certainly the work done in sheep, I think with Haemonchus contortus, has shown that actually by the time you start to see a reduced efficacy as measured by fecal egg count reduction testing, a large proportion of that parasite population have already got resistant alleles to that particular drug. We don't really have any early markers that can help us and support us in detecting resistance early, which would then allow us to change the management and the products we use. There's not much known about the molecular mechanisms of resistance, except perhaps for benzimidazole, whose targets are beta-tubulin genes, and there's been several nucleotide polymorphisms that have been associated with a reduced sensitivity to BZs in sheep and cattle, and the work we've done ourselves in horse nematodes, but we haven't developed any sort of molecular assays for that, because actually by the time we started working on that, it really was too late for BZ resistance in small red worms, for example. Work in horses and other species has also shown that reversion is unlikely, so that if you leave a population of horses unexposed to a particular compound for a couple of years and then you use that compound again, it might work a bit when you first use it, but actually if you use it a second time, the resistant alleles seem to come to the fore again and the product doesn't work. There's a complicated aspect that some of the compounds, particularly the macrocyclic lactones, have a similar mode of action. So the question is, well, if you induce resistance to one type of compound, is that resistance or lower susceptibility then mediated against the related compounds that have the same target molecules? And what they've even found in sheep nematodes is actually not just are the target molecules changed in resistant populations, but what these resistant worms seem to do is to be able to upregulate different proteins that pump things like anthelmintics back out of their body. So they've become quite sort of super resistant. It's not just an allele change at the target, it's other mechanisms which allow these parasites to survive in the presence of the drug. And as I mentioned, there's no new actives coming to market. So that's really the background to why we should avoid blanket treatments in horse populations. And actually, the work that underpins the reason that we don't need blanket treatments has been there in the public domain for very many years and decades, because we have known for a long time that within a group of horses and humans and sheep and cattle is in the case of helminths that although all the animals have potential to carry worms and contaminate their environment, within a group of animals, most actually have low to moderate burdens, and there's only a few tend to have very high burdens of worms, and this is called the negative binomial distribution, and we see it time and time again. And what we tend to find is that horses that are on well-managed paddocks, adult horses, have a very good negative binomial distribution with a few parasites in most horses and only a very small number of animals contributing to the contamination. But we can start to sort of dial that back. So if you've got very contaminated paddocks, particularly where younger horses are involved, we tend to actually see more animals contributing to that paddock contamination, and a higher proportion of them having higher egg counts or test results. So it's really important to understand that the basis of these distribution is how we manage the horses and how we control the level of contamination in their environment. Now, I'm just going to quickly show you some data that it was years and years ago that we did this study where we went on to thoroughbred breeding farms in the UK, and they had at that point, they were not using fecal egg count directed treatments at all. We assessed that by questionnaire, and we took a subpopulation of the 70 farms that we looked at, and we looked at the eggs present and the distribution of eggs in these farms, and we also looked at the type of eggs, and we also did efficacy studies. And what we found on the farms that at that point were very regularly giving ivermectin was that actually when you did egg counts, in this case, I've got other slides with over 1,000 horses, but anyway, this is obviously these results from the first part of the study. Actually, what you find is that a very big proportion of the horses, and in this case, it's mostly adult horses, but not totally adult horses, a big proportion of them have very low egg counts. In fact, in this case, because we're using the modern centrifugal flotation method, the majority of horses had egg counts of under 20 eggs per gram, and then there was just a couple of horses that had egg counts in excess of that level. And when you started to look at these fecal egg count distributions with age across the breeding farms, there was no surprise to find that in the cases where we had higher egg counts, it was much more likely to be found in the younger group of animals. So we did have some yearlings, 2-year-olds and 3-year-olds in the population, and you can see that their mean egg counts are much higher than those of the animals in the older groups. And then we start to think of these more ancient horses, whether they have more susceptibility to strongyl infections than the sort of animals between sort of 5 and 20. So when we're thinking about managing horses, we really always have to think about these high-risk individuals that have higher burdens, they have a higher risk of clinical disease, but they're also the animals that will be the super shedders, the ones that are contaminating most of the environment. But I'm starting to not say so much about the 80/20 rule because we've been doing much more research on this recently, and we realise that actually the level of contamination in the environment, as I said a minute ago, is a real driver for the level of burden in individual horses, as you would predict, even adult horses, and a driver of egg counts. So what we've done in this study is we took 5 yards from practice. We're working with Bell Equine down in Kent, and we identified a low risk yard that had good management, dung removal, regular dung removal, no harrowing, and they did a bit of grazing with, in fact, it was deer that grazed some of the paddocks. And then we had another yard where they had sort of two groups of horses actually. One group where they had dung removal and no harrowing, and another group of horses on the same premises where they had no dung removal and harrowing. So that was a mixed risk population. And then we had 3 premises that were really quite high risk with a high stocking density, no dung removal and harrowing, just very simple study. And we looked at their diagnostic test results. And this is the fecal egg counts that we show here. And what you see as you increase your risk, you lower the percentage of animals that are below 200 eggs per gram, as you would expect, but it's quite substantial. And what we found actually in these high risk populations is actually the old negative binomial distribution really fell apart. And what we found was in some places, almost 50% of the horses were contributing to 80% of the eggs that are going on. So that means that if you went in and looked at these horses to treat, you would always be treating more animals because more of them would be above our threshold that we would assess either as 200 or 500 eggs per gram or a value in between. So it's really important to remember where management is poor, don't make the assumption, and these were all adult horses, don't make the assumption that the negative binomial distribution will apply. So that makes it quite difficult for people in practice and people, owners and yard managers, in trying to get the balance right here. Because what we don't want to do is to have horses with lots of large worm burdens that might have either subclinical or clinical effects on those individuals. But at the same time, we don't want to overuse the anthelmintics to kill those worms, because we know that resistance is a big threat and it's already present, and we don't have any new wormers coming to market anytime soon. So we've really got to think not statically about worm control. We've really got to go in and do a proper risk assessment at each yard that we work with or each premise that we work with to understand the level of pasture management and therefore how good or bad it is, and therefore the level of contamination that's likely to be there, and therefore that will have an impact in driving transmission. It will drive the level of infection in individuals. And when you go to test those animals, you will find that they require more treatments. And what you really need to do is work really hard with these people is to try and drive them away from this, because this isn't, this, if resistance is there and there's no products coming available, this is not going to change. So we need to drive them on this slide so they've got better pasture management, so we have lower levels of contamination, so that when we do go in and test those animals, we don't need to treat as many, and therefore we hopefully reduce the selection for resistance within that population. Oh, it's heating up. Lorna's taking her jumper off now, so it's just getting, this is getting too exciting.
It
is. So anyway, so I'm just going to do a little bit on anthelmintic resistance, just a bit of a background on the published studies that are there, and then talk a bit more about dealing with it in practice. So you're probably aware of all the things I'm going to talk about, but this is just a little refresher. So if you look across the different types of anthelmintics and classes of anthelmintics, we know that resistance is a major problem in small redworm. It's really common to fenbendazole, it's pretty common to parentel, and it's emerging to both ivermectin and moxidectin. In the last couple of years, we've now got reports of resistance in tapeworm to both praziquantel and a double dose of parentel. And I'm suspicious of a few farms, at least in the UK, that have issues, resistance issues in tapeworm. We haven't detected resistance in large redworm yet, and the reasons for that aren't clear, but that's something that we need to keep an eye on. And then for ascarid infections and foals, what we find is a sort of flip of what goes on in small redworm. We find that resistance to the macrocyclic lactones, particularly ivermectin, is really common in Parascaris, with resistance reported in pockets for both fenbendazole and parentel. If you look at the cyathostomin situation in a bit more detail, it really is quite depressing. This was a really nice study a survey of the data, of the published data by Martin Nielsen that was came out a few years ago. And what he did is he looked back to 2000 and he looked at all the reports that had been published for resistance to the various compounds, in this case against small red worm. And what he found was that the 58 studies reported there was 100% prevalence of resistance reported to BZs. For Pyrantel, fewer studies, but 92% of those studies reported resistance to Pyrantel in small red worm. And for macrocyclic lactones, there'd been about 57 studies undertaken, with 23% of those studies reporting a reduced effectiveness of macrocyclic lactones. What he found in the macrocyclic lactone studies, that most of these reports were from South America, where there might be questionable generic compounds available, but there was two more recent ones in the US, and that was associated, very embarrassingly, with horses that had actually been imported from stud farms in Ireland across the United States, and they'd carried with them their ivermectin and moxidectin resistant cyathostomas to the next stud farm. So if there's nothing else you take away from this talk tonight, is remember to do quarantine procedures when horses are moving from site to site, because it's really important, especially where resistance is a threat. The other method of looking at reduced effectiveness of anthelmintics is to do have a look at the strongyl egg reappearance period. And basically that is a useful monitoring tool, particularly for researchers like myself, for assessing whether or not resistance or reduced susceptibility might be present within a population. And what you do in that case is you look at the number of weeks after anthelmintic treatment until the fecal egg counts become positive, or in a better case, actually reaches a specific threshold. So you would look at the egg counts and say, well, actually, when do they become less than 90% effective? Now, I'll show you some data in a minute, but keep in mind that the egg reappearance period, just on the basis of either looking at a threshold or looking for the presence of eggs, for ivermectin, when it first came out, was between 9 and 13 weeks. And for moxidectin, it was originally around 12 to 24 weeks. That was a really suppressive anthelmintic moxidectin at reducing and suppressing egg counts with strongyl eggs, probably because of the pharmacokinetics of the drug, in that it's very lipophilic, and then it slowly comes back into the plasma and maintains these levels, which would then suppress maturity of the parasites and egg production. And it's important to keep in mind, though, it's quite bad news for both these compounds, because there's numerous studies reported for a shortened egg reappearance period for macrocyclic lactone against strongyls. I think actually, if anyone looked at that now, probably, unless in very rare cases where horses hadn't been exposed to treatments for the drug, I think most people would find this. So this is way back 12 years ago. We reported reduced egg reappearance period, reduced strongyl egg reappearance period in young horses on those stud farms that I mentioned earlier, that had been overusing macrocyclic lactones for years. We were detecting eggs after the treatment of foals and yearlings within 2 weeks in some cases, and 4 weeks. And then we reached a reduced less than 90% reduction threshold between 6 and 9 weeks. And it's not just a problem on stud farms. This is a more recent study that was actually led by Tim Mayer a couple of years ago, where we looked at a large number of leisure horse yards, and we found that the percentage of horses with a fecal head count reduction of less than 90% was really, after, and this was moxidectin treatment, was really starting to increase, and it was over 20% within 12 weeks. So these egg counts are coming up quite quickly. And of course, the temptation is to go in and treat again, but that's something that we really need to think about and model, about how soon we should treat horses and how soon we should egg count them post-treatment and how soon we should come in with treatments. Likewise, as I mentioned earlier, resistance is common in Parascaris, and it's a particular problem with macrocyclic lactones, but there have been reports of resistance to Pyrantel and BZ in this worm. And in fact, there was a combination product that was tried out on farms in New Zealand, but over 10 years ago now, and they already found reduced efficacy of a combination compound containing ivermectin and fenbendazole against Parascaris on one of these farms. So I don't think combination products are the solution. I don't think they help. Our friend, the pinworm Oxyurus equi, doesn't want to be left out of the equation either. And although there's been far less studies performed and it's technically more challenging than a standard fecal egg count reduction to look at efficacy against this worm, in the few studies that have been undertaken, quite commonly macrocyclic lactone resistance has been suspected or identified. And as I mentioned in the previous slide, although it's been far less studied, and it's particularly challenging because fecal egg counts have such poor sensitivity for detecting tapeworm infection, people are starting to look at this now, including ourselves, because of the concerns of clinical cases on farms where praziquantel in most cases or parentel have been used. And Martin Nielsen took the opportunity to look at this in farms in the States a couple of years ago, and what he observed in terms of fecal egg count reduction was that it was only reduced by 23% following praziquantel treatment, and over 50, just around 50% with parental treatment in the same groups of animals over time. So he therefore reported a sort of double reduced efficacy of both available compounds for tapeworm. And this wasn't just an issue in youngsters, which are likely to shed more eggs anyway. He actually looked at the effect of praziquantel in mares, and he found that out of 84 mares that are positive for tapeworm eggs before treatment, 7 of these still excreted tapeworm eggs after treatment. So although we haven't got a validated test for fecal egg count reductions for tapeworm, finding cestode eggs 2 weeks post-treatment, particularly in multiple animals, is a significant finding. So how do we address resistance in practice? Well, first of all, we've got to look for it. There's no point in assuming it's there, and there's no point in assuming it's not there. And this is supported by several questionnaire studies and surveys that have been done, including our own, where if we go out and ask people, particularly horse owners or managers of farms, are you worried about resistance? They say yes. Are you worried about it on your own farm? They say no. And then we say, have you looked at it? And oftentimes they'll say no. So people know that it's a sector problem, but they don't really want to look at it in their own backyard, literally. So it's important when you're going out and talking about parasite control, you've got to start saying to yourself and to your clients, is this wormer going to work? Well, actually, I need to test that. So that's the first thing we need to do. We need to think about looking for resistance and if it's there. And at the same time, regardless of whether once you've done a fecal egg count reduction test, if it's there or not, you have to keep advising people to avoid those practices that select for resistance. Try and avoid all group treatments. Make sure you weigh animals properly before you give them a dose, and certainly don't use the wrong product in the wrong host by the wrong route. And what you need to do, as well as I'm telling people to avoid those practices, is to promote practices that will help us reduce the need to administer anthelmintics. And that includes using management tools in the environment to reduce transmission, and the use of diagnostic tests to identify the animals that are infected, and to identify the animals that haven't got a particular burden of worms, to reduce the frequency of treatments that we apply. And the better management that is instituted at that particular premise, what you'll find is the less animals require treatment based on the results of diagnostic tests. Let me quickly touch on this because I'm sure everybody knows this. Is the management tools that we have, they aren't rocket science. So what we recommend is people remove dung at least once a week. Horses grazing outside year round, they should be doing that year round. Try and graze animals at a stocking density of at least 1 acre per horse, and try, if possible, rest or rotate pasture with a minimum of 6 months between the groups of horses. And ideally, to rest that paddock in the first 6 months of the year, because what we know, particularly for strongyle nematodes, is that the parasites that are put out in one season, they will overwinter, but what they will do because of the restricted food reserves is they'll tend to die out by the following summer. So if you leave those paddocks free of horses that won't recontaminate those paddocks, by the time you get to summer in the second year, most of the worms from the previous year will be reduced. We don't know the specifics about the persistence of ascarid eggs and tapeworm in mites, for example, but the longer you rest paddocks from horses, by default, the lower the contamination with those infective stages will be, because they don't live forever. Co-grazing with ruminants or alternate grazing with ruminants is also a good idea, particularly during that rest period, but keep in mind to do a risk assessment for liver fluke in any ruminants that are co-grazing or alternately grazing with horses. The super tool that people use, and many people use this now, and it's great to see it because nobody was doing it before the 2000s, is to use egg counts to direct treatments. And we're using these egg counts to direct treatments to stop shedding. We're not really doing it to stop the burdens on horses, although that's a side effect of the treatment. We're reducing burden, but really we're targeting those treatments to reduce egg shedding and to reduce contamination into the environment, with the hope being that within an adult population on good grazing, what you'll find is that the vast majority of horses will have a low egg shedding level and you don't need to treat them, and there'll be a few horses that you do need to give anthelmintic. And then what you hope is that any resistant eggs that survive in the treated horses that develop to L3, those L3 will then go into the paddocks and they'll mix with this refugia, these other parasites that are either already on the paddock or are being produced by horses that have not been treated and therefore not been through a round of selection. Now, fecal egg count reduction testing is something that everybody should be now putting into their annual worming plans, and they're really simple to do. So basically, you just do your egg count. I would say ideally in the summer because you're more likely to have more horses positive with a higher EPG in the summer, and therefore you can include more horses in your efficacy test, and it will be more arithmetically or statistically robust. So you test your horses, you identify the horses at over 200 eggs per gram, and to save you having to recount them again, I would then treat them very soon after that egg count's done, and then 2 weeks later, take samples to calculate the mean reduction in egg count between the sample taken just before treatment and the sample taken 2 weeks after treatment. And as I mentioned earlier, you would be expecting susceptible parasites to have a reduction of more than 90% for Pyrantel and fenbendazole treatment, and over 95% after ivermectin and moxidectin treatment. And these are just some examples of the data that you can generate. This was that Thoroughbred Stud Farm study from over 10 years ago, where we went in and we did fecal egg count reduction tests on a number of the farms with a number of products. This is all Strongel egg count data. And what we found was that fenbendazole really wasn't working at all. In fact, we stopped using it for ethical purposes because in some cases the egg counts were increasing. And then when we tested Pyrantel, we found a bit of variation in efficacy depending on the farm and depending on the group in the farm. And what we tended to find was that young animals, the efficacy appeared less, and that's probably because they've probably got higher burdens to start with, and the pharmacokinetics might be different in these younger, thinner animals. And then when we looked at moxidectin, we thought that doesn't look too bad. We've actually got 100% efficacy 2 weeks post-treatment. Keep in mind, this was done 12 years ago. And then we identified a reduced efficacy on one of the farms with ivermectin. And then we did a similar study with leisure horses where the pressure doesn't seem to have been so high, but again, we got ubiquitous fenbendazole resistance. All mean reductions in egg count were well below the 90% threshold. We found a better effectiveness of Pyrantel on many yards. So again, this is something that Pyrantel should be there in your toolbox, but keep in mind you should check its efficacy by doing fecal egg count reductions. And we found similar results with both moxidectin and ivermectin in that all the yards actually in the study had a mean egg count reduction of over 95%. So these are the tools that we should use in practice. These are just examples of what you can do just to get an idea of the drug sensitivity in the worm populations on site. Now, the other tools that we can use in practice are those that we use as diagnostic measures to target horses for anthelmintic treatment. Now, as I mentioned earlier, fecal egg counts have very low sensitivity for detecting tapeworm, and that's partly, it's not just to do with the methodology and there's intermittent release of eggs from proglottis into dung, but also associated with the fact that many horses that have tapeworm infections have a high proportion of immature worms or sterile adults, so that these don't release eggs. So you would detect them by a standard or even an increased volume fecal egg count test. The other thing about tapeworms is that because the proglottis sometimes don't rupture, either until near the end of the GI tract or once they're in the fecal pile, you find that the eggs are concentrated within the fecal pile, and that can really confound the results, even when the samples are quite well mixed. So what's been developed over the years was initially a serum-based ELISA for tapeworm infection, and subsequently a saliva-based ELISA. And both of these tests are based on the measurement of IgG-T antibodies to purified 12 to 13 kilodalton excretory secretory antigens from Anaplacophila perfoliata. And work done with both the serum test and the saliva test has shown that these antigen-specific antibody levels show strong positive correlations with the level of tapeworm burden. And in so doing, if you measure antibody, you can therefore infer the level of that animal's worm burden. Now, we do know that Anaplacophila perfoliata burdens can be associated with intestinal damage and therefore colic, and that's been associated with burdens over 20 worms. So it's important to keep that in mind when you're looking at groups of animals that you're assessing in the test, because both the tests that are available through Austin Davis now, the serum test and the saliva test, all horses that fall within the borderline and moderate high categories in the validation studies, all horses that had over 20 tapeworms fell within those categories, and that's why we advise treatment for all horses within the borderline and the moderate high categories that you get with the results of the test. And there weren't any horses with more than 20 worms in the low category. That comprised horses that had no worms or worm burdens less than 20. So you can use both of these tests to identify the horses that will contaminate paddocks, and you can also identify horses that are at risk of colic if they've got a higher burden. It's important to remember, because the antibody-based tests, is not to do them too soon after treatment, because then you risk measuring antibodies that were present at the time of treatment to the worms that were subsequently killed. So we recommend that serum test isn't done until 4 months after the last treatment, and saliva tests are not carried out until 3 months after the last anthelmintic treatment. We recommend tapeworm testing in spring and autumn for the following reasons. So we know that in spring, when you look at studies that have been done in abattoir surveys, you find that this was in Italy, this study, that obviously animals accumulate tapeworm burdens over the summer, and they start to mature and increase, so they're at high levels in the autumn, and then the worms slowly mature, so that by the time we get to spring, the highest proportion of the parasites are adult worms. So this is an important time to think about targeting treatments using diagnostics to identify horses that will have adult tapeworms that will be likely to contaminate the paddocks with eggs at a time when it's epidemiologically important in terms of mite development. It's also important to consider to look at testing horses in autumn, because this is likely when they've had accrued their highest worm burdens and therefore more risk of disease. Now, for low-risk populations, we can recommend that you can test in spring or autumn, but you have to do a good risk assessment for this, and also ideally look back at previous ELISA test results to confirm that most of the horses within the population do have low serum or saliva scores. In other groups that are moderate to high risk, we recommend that you test in spring and autumn for tapeworms in order to reduce egg shedding at the start of the grazing season when it's starting to get warmer, and to reduce burdens of tapeworms at the end of the grazing season to reduce the risk of clinical disease. Now, this is backed up by some recent data that we've just published in International Journal for Parasitology, where we worked with collaborators at the Royal Veterinary College, where we looked at the number of mites recovered from 3 paddocks across 3 yards down in the south and east of England. And what we found is the eribatid mites that were hoovered up from plots on these 3 paddocks actually changed their numbers significantly over the season, so with much higher levels of mites starting off in the spring and increasing and then reducing again in the autumn, and then start to increase again in the spring and maintaining levels over the summer. And that's why it's important to start to look at horses in the spring for tapeworm testing, because what you want to do is identify egg shedders at this time of year to reduce egg shedding, so that you don't have this interaction with the mite when the mite levels are increasing on paddocks. And we took this on a step further. So what we did is we extracted DNA samples from the mites that were obtained. We did it in different mite families, and this is the amalgamated data here. And then once we extracted the DNA, we took pools of mites from the DNA samples and we subjected them to PCR with Anaplacephala specific primers. And what we found is that, again, looking at our DNA samples that contained PCR products that were positive for Anaplacephala infection, we found again the sort of seasonal influence with these very big increases in tapeworm positive mite DNA samples, particularly going on from late spring into summer. I mean, I was really quite shocked. These are quite prevalent farms, but on what and the samples taken, for example, in April, there was over 90% of the samples assessed, and you can see it's quite a lot of samples assessed. There was over 70 actually had Anaplacephala DNA. So you want to break that life cycle. You want to come in and think about testing in the spring, reduce the egg output so you can start to plateau off the level of infected mites by reducing contamination onto paddocks. And this just shows you the data set from Equisal, for example, that actually using tapeworm testing can significantly reduce the number of applications of anthelmintics, in this case, obviously, pseudocidal anthelmintics. And out of the over 200,000 saliva tests that were run between 2015 and 2023, only 1 in 3 horses were recommended for treatment. So that's a big saving in anthelmintic. This just shows you the typical sort of yard prevalence that we get across yards, and actually most yards in the UK have relatively low levels of tapeworm infection as indicated by testing, so you won't be using a lot of anthelmintic in those yards. And then we have the occasional yards where there's high levels of transmission and prevalence, for example, up to 90%, and it's on those yards that you really have to start giving advice about improving management and probably more regular testing to find those animals that are contributing to contamination that will then infect the rest of the group. The other tool that you can use to reduce anthelmintic treatments is a small red worm blood test. I won't mention much of this because I think we're going to come back and talk about this in our next session, but this is a good test to use in low risk populations of horses, and you can assess the risk level using various tools, including What's Your Worm Risk, which is a free online tool that we've developed for people to try and help them ascertain the worm risk of individual horses. And in those low risk populations of horses, you can actually use the small red worm blood test to identify horses with very low burdens that don't require treatment. And I'll come back to that at our next session. So that leads to me on to the next thing. So we've talked about resistance, we've talked about the worms that are there, we've talked about the tools that we can use within our armoury that can help us both detect resistance and to reduce anthelmintic treatments to reduce selection for resistance. So what do you do if you do monitor and you discover it on site? And we can talk a wee bit more about this at the end, but really you've got to identify what's there, the level of efficacy, and against what parasite. And if you do detect it, you have to think about what you can do going forward for that site. The first thing you've got to do is you've got to reduce further selection. There are no new compounds coming to market. You've got to preserve the efficacy of the ones that you've got to do available to use on that particular site. And the best way to do that is to improve management, to reduce transmission, and therefore come in with testing, and then hopefully, if the management improvements are successful, you will find that over time, less horses will test positive and therefore less anthelmintic treatments will be required. And in that way, you're actually getting on top of what's going on at that particular site with data, and you feel a bit more assured that what you're using is actually working. And if your test results start to increase again, the number that are positive, then you have to work out what's going on there that's going wrong, whether it's the management is lapsed again or whether the products that you've maybe changed to or continue to use are actually becoming even less useful for reducing worm burdens. Has anyone got any questions at this stage? Because I realise I've talked constantly for 25 minutes now.
Anyone, any questions? Feel free to put anything in the chat as well if people want to, if it's easier. No? I take it that's a no. No, I don't think any questions at the moment, Jacqui.
Good, fine. Okay, so we had some questions that had come to you from your members that you put to me. And I don't know if those people are here tonight, but if not, I hope they watch the recording. So one person asked, how do we choose the appropriate wormer for each individual horse? Well, obviously you don't pluck that out of the air. The first step you have to do is to do a risk assessment to identify whether that horse is at a low risk or higher risk of infection. And then you've got to consider your knowledge of the epidemiology of the parasites to consider which worms that horse might be at risk of. Is it parascaris? Is it small red worm and or tapeworm? And then you can then use diagnostics to inform your decision about what you're going to treat that horse with. So, for example, if it was a horse you identified as low risk and it was the autumn time, what you could do is say, well, actually, I could withhold treatment, but there are certain risk factors that I'm not sure about, and the owner's really keen to treat the horse. What we could do is we could test it with Equisal, for example, and we could test it with a small red worm blood test, or we could do tapeworm and red worm blood tests. And what you might find in that horse, that say it was positive for tapeworm and negative for small red worm, then you would give it a tapeworm treatment such as praziquantel would be the choice, rather than putting a selection on the nematode by using Pinantel. And then, for example, if that same horse was positive for both small red worm and tapeworm, then you would consider whether to give it a combination product or whether to give it Pinantel based on your knowledge of the effectiveness of that worm, of those wormers against the different types of parasites. And then if it was positive for small red worm and negative for tapeworm, then you would give it an anti-nematode therapy, and then you'd have to consider whether it needs larvicidal treatment if it's not at risk of larvicide, high larval burden. You could then choose another small anti-nematode treatment, such as ivermectin or Pinantel, if you were assured that the Pinantel would work. So it's not a sort of like just one-step process. What you've got to go through for each individual horse is the risk assessment for that horse based on its age and signalment, based on its management, and ideally based on the results of diagnostic testing, including egg counts as well, which I didn't mention because I was talking about autumn there. There are some instances where you might need to come in with strategic treatments, and I'll come back to this in a minute, and that would be foals at high risk of ascarid infections, particularly foals less than 6 months, and young horses that are at a risk of significant burdens of encysted cyathostomin, so that would be horses between 1 and 5 years old, particularly if they're on poorly managed paddocks, and then you would consider giving them amoxidectin treatment. So I hope that answered the person's question. There's a whole chapter on this, if you haven't already seen it, in the canter guidelines, which are accessible through canterforhorses.org.uk, and there's a whole chapter on selecting and using anthelmintics appropriately. And the chapter before that provides you with information on risk tables and assessing risk, on which the whatsyourwormrisk.com calculator is based, because it was Tim Mayer and I developed the whatsyourwormrisk.com calculator, and we also did the risk tables for canter. I know people get a bit stressed if they think, oh, well, it's all about what we do when, so I'll put it down in a calendar here so it becomes a bit clearer for people. So the first thing to always think about is to risk assess, try and work out what that horse is exposed to and its level of susceptibility to worms. Make sure that the best sort of paddock hygiene has been applied as possible. If not, try to get the owners to improve it. And then you can come in with testing. So if horses are out all winter, for example, or out for part of the winter, and they haven't been treated over the autumn or winter, or they received a treatment in the autumn or early winter, you could come in and do a fecal egg count in the sort of late winter time, late February, early March, just to see what the egg counts are like, to get an idea of what shedding is and if any require treatment. And then moving into spring, you could then do another egg count, straw gel egg count, just to check which horses are positive that require treatment to reduce egg shedding, and you can apply a tapeworm test at this time to identify any horses that might be excreting tapeworm eggs, particularly because this is a time of year when the mite levels go up on paddocks. And then you can do another fecal egg count in the summertime, ideally at least 8 to 12 weeks after your last egg count, depending on which product you use to treat the horses that were positive. And that's a good time of year, as I mentioned earlier, to test for efficacy. And then going into the late summer, you consider another egg count, particularly if you've used a shorter acting product in the summer, just to check the levels of contamination going on to paddocks, particularly if horses are going to be staying out for a significant time. And then coming into the autumn time, you have to think about your tapeworm tests to reduce burdens, to identify horses for treatment to reduce burdens that might be pathogenic. And then you have to do your risk assessment to work out whether or not those horses should have a larvicidal treatment, whether you don't want to treat them at all, if you've got a really good risk assessment and lots of repeated egg counts that are very low or negative, or to apply the small red worm blood test to inform whether or not they do require treatment. Was that clear? Yes. Now, this was a funny question. I had no idea how to answer this. How do you deal with herds of thoroughbred weanlings and 2-year-olds? Surely that's a veterinary clinician's problem. So what do you do? Well, I think handling should start early. I mean, especially if they're thoroughbreds, they're going to end up doing something fancy-mancy on a race course or as a sport horse or something. So I guess early handling is a good thing, as shown in this picture. And then I suppose you've got to be pragmatic about it. So you've got to think about what the practical options would be for that group of horses if they were completely crazy, whether you should consider strategic treatments, but how easy is that going to be? So it's really important to get people to be able to handle these horses and allow them to be wormed if they're at risk of disease. You have to think about testing options. So fecal egg counts, for example, if they're not handleable, you could have someone stand in the field all day watching them, or you could find a way to bring them into an enclosed yard or into the box with the mare, for example, and then you can pick up the poo fresh from the identified horse. Blood and saliva tests obviously require some sort of fluid from the horse to do the ELISA. We find a lot of our stud farm vets do blood tests actually rather than the saliva. I think they just find it easier from a management point of view. They're just going to line them up. They might be bleeding them for something else rather than bringing them in for the half hour, not feeding them, tying them up, not giving them access to something to drink before they do the equisal saliva test. So we do find quite a lot of the big farms. We're really busy in tapeworm serum testing at the moment because a lot of the big farms are testing at the moment. And then, of course, always emphasise the importance of testing all animals in each group because of that negative binomial. Don't just say to people, oh, just do a couple of them and you'll find, because chances are you might get all the ones that might be low and then you say, well, no treatments are needed, and actually you might miss a few animals that really do need treatment. Or on the flip side of that, you might pick some of the ones that need treatment, but there's a whole lot of other horses that don't require treatment. The other thing I was saying, I see this now, I look at our stud farm data quite a lot, is the adult horses really act as excellent sentinels of helminth transmission at sites, at these farms, for example, and diagnostic testing of these groups is important. So I know I can look at some farms where I know the management must be brilliant because the mares, although they're grazing with youngsters or they're grazing paddocks recently vacated by youngsters, their serum scores are really low. So that must be very good paddock management because the mares aren't getting exposed to the worms. So they're acting as sentinels. In addition to providing advice about whether or not the mares need treatment, it's given really good sort of immunoepidemiological information on that site about how good the management is. Sadly, sometimes we see that and then we see high tapeworm scores, and that's another story for another day. Okay, and the final question, before my nose completely explodes, is advice on best practice management for young stock and studs. So I have written some articles on this, which I'm happy to share with Lorna if anyone wants information on that. I've got lay ones, and I think I might have some sort of veterinary directed ones. And this is actually also there's a chapter about to be published in Cancer Guidelines, not written by me, but written by others and reviewed by the group, that's going to be put on the website in a couple of weeks. So there will be further information on this. But again, the best practice management is the best practice management as it is for everything. It's like reducing transmission by good paddock hygiene, proper quarantine, low stock and density, no harrowing if possible, certainly no harrowing if horses have recently been on the field and poo hasn't been lifted. And then thinking about those worms that might be a threat to horses of different ages. So obviously there's strong galloides, but I wouldn't recommend treatments for that unless you really, really were suspicious that it was causing a clinical problem in young foals. So the next worm you have to think about are the ascarids, particularly foals up to 6 months old, where particularly on farms where there's likely to have been high levels of transmission, you want to come in with strategic treatments at 2 and 4 months against ascarids. And then as they get older, they'll switch over to having strongel infections and strongel eggs in their fecal samples. So you can start to use fecal egg counts at that stage to direct your type of worm that you use across the summer. And then as these horses, particularly if they're older foals, if they're nearly yearlings coming into that first autumn or in the next year, you've really got to consider them at risk of large larval cyathostomin burdens, and then you would institute an anthelmintic treatment against those stages. I would usually always reach for moxidectin unless you've got proven efficacy of fenbendazole on that particular population. And then again, from 6 months old, and again, adult horses as well are susceptible to tapeworm infections, although probably younger animals are more susceptible to higher burdens of disease. Then you have to consider testing for tapeworm and treating accordingly. And then at the back of your mind, you're probably thinking about large redworm, particularly if you really reduce the amount of treatments on a particular farm, and particularly if that farm's an open herd. You can do Westgate Labs now offer culturing of large redworms, so you can screen for that. Pinworm's an obvious thing and a menace, which you usually have to deal with at an individual level. And then there might be occasions where there's horses, donkeys on the farm, or there's co-grazing with sheep or cattle. You have to think about liver fluke as well. So in terms of special considerations for foals, as I mentioned a minute ago, you've got to unfortunately come in with strategic dosing for those young foals. And at the moment, we're recommending to worm these with about 2 or 4 months with either fenbendazole or pyrantel, unless you know it's a really low risk foal. Perhaps it's one foal grazing with a mare or a couple of foals at very, very high acreage. And then from 6 months, you can fecal egg count test these foals, and you can look to see whether or not they've got ascarid eggs or strongel eggs or both. And then you can select the wormer based on the egg species that you identify by the fecal egg count test. Again, it's important to assess efficacy of the products against both, ascarids and strongel. So I would think that first fecal egg count test that you do, if you have a lot of positives, it'd be really good if you could do a repeat test 2 weeks after treatment. As I mentioned a minute ago, in your younger animals, particularly the older foals and in your yearlings and 2-year-olds, you want to be thinking about larvicidal treatment in late autumn. And particularly in yearlings, going into 2-year-old year, we quite often find that they're all grazed together and they're out all the time. Sometimes their egg counts are going up quite quickly over the winter, and you might want to do either a second egg count in sort of later winter to try and work out what's going on. In some cases, you might want to come in with a second larvicidal dose if you're concerned that the transmission has been high over the winter. And then you can start to test tapeworm from 6 months. Remember, not to use saliva in non-weaned foals, because the mare's milk contains antibody that will then lead to false positive test. And it's super important to optimise your environmental control measures on foal paddocks, especially with respect to ascarids. And please do advise people to not use the same nursery paddocks year after year, because that's a bit of a recipe for disaster in terms of build-up of ascarid eggs in the environment, leading to over-treatments, which will then lead to resistance. And then for other youngsters, then you probably in summer, you just treat them like other groups of horses. You do fecal egg count directed treatments. You might find that you have to do them more frequently because the egg appearance period can be shorter in young animals because they tend to have a lower immunity and accumulate higher burdens quicker. Because these tend to be higher risk groups, I'd recommend you take worm test them twice a year unless repeated results indicate a very low prevalence within that group of youngsters. But be aware of emerging resistance and tapeworm. That I think is going to become an increasingly significant problem, particularly on large farms. Again, small redworm larval stages are undetectable, but these are high-risk animals, so we would recommend a larvicidal treatment in the late autumn, early winter. And as I mentioned before, you may need that second treatment or egg count later winter, and also be aware of possible resistance to MLs, especially as expressed as a reduced egg reappearance period. And that's me. You will be glad to know.